The process of meiosis is part of a larger growth cycle of a cell. So we said that the life cycle of a cell was from one cell division to the next. Within that life cycle, we have several really important phases that lead and prepare the cell for cell division for next time. This cell cycle is controlled by a variety of checkpoints that limit when and how the cell can actually progress through its cycle and through cell division. So this is important in controlling how often a cell divides, and it's also important in controlling cell division in the sense that the cell should not divide if it's not ready to divide. So each of these checkpoints is a stage with a very specific end that allows the cell to determine if it's ready to move on to the next phase in its life cycle. So the main part, or the majority of a cell's life, is spent in interphase. Interphase was named originally because they thought that during cell or in between divisions there was nothing going on. So it was sort of the phase in between cell division. In actuality, there's quite a lot going on during interphase. If you were to think of a cell cycle as being 24 hours, which is pretty representative of, say, a skin cell, most of its life time, about 23 hours, would be spent in interphase, and only about an hour is actually spent in the process of meiosis. A bacterial cell actually divides within a few minutes, so it's even faster. Within interphase, we have several important steps or phases. The first phase, immediately following a cell division, is called the G1 phase, or the first gap phase. So in this first gap phase, the cell is basically working, it's doing its job within the organism or within the environment. It's growing, it's expressing its genes, it's producing its proteins, it's doing all of those functions that we learned about in Chapter 4, building macromolecules and moving products within and out of the cell. After that is the S phase, or the synthesis phase. So during this phase, DNA is replicated. So it's during this phase that we go from our single chromosomes to our sister chromatids. So we'll talk about DNA replication a little bit later. For now, we'll just say that it happens. Following the S phase is a second gap phase, the G2 phase. And in this phase, there's a bit more growing in preparation of cell division, maybe some gene expression doing a bit of its job in the organism. And then if the cell progresses through G1, S, and G2, and it's met all of its checkpoint processes, then the cell is allowed to divide, and it's allowed to enter the meiotic phase, or the M phase of the cell cycle. So the cell cycle is made up of interphase and meiosis. And then within interphase, there are a series of stages. And then also during meiosis, there's a series of phases. They're just not drawn here because there's several of them and they happen very quickly. As part of mitosis, or cell division, we have a phase called cytokinesis, which is where the cytoplasm of the cell divides. So mitosis is basically the division of the nucleus, the partitioning of the genetic information, and that works alongside cytokinesis, which is the partitioning of the cytoplasm and the rest of the cell. Some cells can also enter something called the G0 phase. This is a phase, either temporary or permanently, that a cell can enter where there is no cell division. So some cells enter G0 and stay there forever. Nerve cells are one of those. Once your nerve cell is produced, there is no more cell division. That's it. That's all you have. That's why nerve disorders and brain disorders are so detrimental, because those tissues are not regenerated. They're not repaired or replaced. Other tissues can enter G0 as sort of a holding pattern. So, for example, our liver, once it's formed, it doesn't regenerate, not like your skin cells, not like some of your other organs. So new liver cells are not produced unless the liver is damaged. So the liver in a healthy person with no liver damage may spend their entire life in G0 phase. But if some damage does happen and that liver needs to repair itself, it can come out of G0 and go into the meiotic phase. Cells can also enter G0 if any of these checkpoints along the way are not met. So they can sort of pause their cell division until the problem is fixed and the cell is ready to go. So we are going to focus on the M phase, or the mitotic phase. And as I said, this is division of the nucleus and the genetic information, and then division of all of the organelles, partitioning of the organelles in the cytoplasm into the two daughter cells. So the process of meiosis, I'm sorry, I'm saying the wrong process. The process of mitosis begins with chromosomes condensing, and then chromosomes are pulled apart. And once those chromosomes are pulled apart and the cell's almost done, then cytokinesis will take place. So cytokinesis actually takes place simultaneously with the end of mitosis. So we kind of lump it all into one phase, but cytokinesis is its own separate process. We're not going to talk about cytokinesis very much. We will talk about it a little bit once we get to the end of mitosis, but we're really going to focus on that division of the genetic material, the division of the nucleus in mitosis. So we said that there are several checkpoints in that cell cycle that limit cell growth. It regulates when the cell divides and how often. One of those really important checkpoints before mitosis can actually move on and the process of cell division can actually begin is the formation of a specialized organelle called the centrosome. So the centrosome are these little T-shaped organelles here. You have a pair of centrosomes, or two pairs of centrosomes actually, one pair at either end of the cell. So the centrosomes replicate and then they begin to produce microtubules. And so coming out of them are what we call spindle fibers. Those spindle fibers are microtubules that come out of the centrosomes, and their role is going to be to attach to chromosomes and to move them. But cell division will not take place if those centrosomes don't form and the microtubules don't begin to grow. So that's a really crucial checkpoint. If you remember from chapter 4, microtubules are long, thin rods that can be taken apart and rebuilt. We talked about them as giving some structural support to the cell, sort of like scaffolding. We also talked about them being involved in movement. So vesicles will move along those microtubules to move products within the cell. The third function of those microtubules is to grow out of those centrosomes to move to either pole of your cell. So if you think about this like Earth, you have a northern hemisphere and a southern hemisphere, and you've got centrosomes at each pole, like the North and South Pole, and then you have an equator, and we'll talk about that a little bit later. But those centrosomes are going to move to either end of the cell. Microtubules are going to grow out of them, and they're going to attach to the chromosomes. And then those microtubules are what's going to actually move and partition those chromosomes into the daughter cells. So we are going to go through each of the phases in mitosis. You do need to know what happens in each phase. You need to know what happens in each phase of interphase, and you need to know what happens in each phase of mitosis. Your book has a really good diagram that shows all of these stages and gives quite a lot of detail on what happens in each one. You can find that on page 186 and 187. Also on that page you have pictures of a real cell whose chromosomes have been stained, and then the cartoon version that's easier to see what's actually going on with that chromosome movement. So mitosis sort of actually begins in the end of interphase, so at the end of G2. In the end of G2, those centrosomes duplicate and they begin to produce microtubules. So there, at the end of G2, we have our centrosome checkpoint where we actually check, the cell checks to make sure those centrosomes have been formed. At this point in the cell, we still have all of our DNA sort of spread out and unspooled in the nucleus. We have a nuclear envelope. If you remember, that's the membrane that surrounds the nucleus. And at this point, our chromosomes have been duplicated. They were duplicated in S phase, which comes before G2. So chromosomes were duplicated, so we have sister chromatids here, but they're not condensed yet. At the end of G2, at the end of interphase, we enter the phase of mitosis. The first stage of mitosis is prophase. And in prophase, those microtubules get bigger, they get longer, the centrosomes spread out to the poles, and then our sister chromatids begin to condense. So our chromatin gets tightly packed, our chromosomes are tightly packed around proteins, and we get those densely packed sister chromatids that we saw in the slide in the last video. So at this point in prophase, this is the first stage of mitosis, our chromosomes are becoming condensed. There is sister chromatids, and they are getting ready to attach to microtubules for the next phase. Our next phase is called pro-metaphase, so this means before metaphase. That's the next stage. In pro-metaphase, the nuclear envelope or the nuclear membrane begins to break up. That's really just to make room for those chromosomes to move around and the spindle fibers to attach. If you look at this picture compared to the last one, our chromosomes are much more condensed. So they're really tightly condensed. And the reason for this is if you think about a wad of yarn or a wad of shoelaces, if you just reach in there and try to separate them, they're going to get tangled. DNA is formed with some very strong bonds, but they can break. So if the DNA gets tangled, you're not going to partition it correctly, and there's also the potential of damaging that DNA. And remember, the point of replication and mitosis is to produce cells with the correct complement of chromosomes. So at this point, our centrosomes have reached the poles, our microtubules have grown out, and they have attached to the chromosomes at a region of the centromere called the kinetochore. So you can sort of see where they are attached there. Remember, the centrosome is sort of the pinched in waistline of the chromosome. And I like to think of the kinetochore as kind of like the belt around the waist. And if you think about someone going in and grabbing your belt loop and trying to pull you by your belt loop, that's kind of what a microtubule is doing. It's attaching at that waistline and it's going to pull a chromosome one way or the other. After pro-metaphase, that nucleus is gone. And the chromosomes are all attached to their microtubules. And in metaphase, those chromosomes actually line up along the equator of the cell. So if we think about this like Earth, we've got a line down the center, or across the middle, not down the center, but across the middle, and those chromosomes are lined up with one chromosome facing each pole. And this is really important so that the chromosomes are separated correctly. Remember, we want one copy of each chromosome in our daughter cells. So the chromosomes have duplicated in the S phase. They are attached at the centromere. And now they are going to be pulled apart. One copy of each sister chromatid to either pole. And so metaphase is really important in lining up those chromosomes and making sure that one chromosome of each of those sister chromatid pairs is separated into each daughter cell. So at this point we have another important event. Those sister chromatids were not really just held together at the waist with sort of arms flapping all over the place. They were actually held together almost like a zipper by proteins called cohesins. So at this point, the cell will break down those cohesins, those zipper-like proteins that are holding these proteins together. And once all of those cohesins are broken, those chromosomes can be pulled apart. So our next phase of mitosis is anaphase, and this is one that's really easy to see on a microscope. Metaphase is pretty easy as well. Anaphase is really quick. Once those cohesins are cleaved in metaphase, the process of anaphase begins, and those sister chromatids are pulled apart, and they're pulled to either pole of the cell. So at this point, when anaphase happens, we now have doubled our number of chromosomes temporarily in that cell until the cell pinches into two. So from our sister chromosomes, we have separated daughter chromosomes. And then those microtubules do two different jobs at the same time. You may have noticed that some microtubules are attached to chromosomes. Other microtubules are attached to each other and extend the length of the cell. So we can see a few of them in here. Those microtubules that are attached to other microtubules actually elongate at this point and sort of stretch out the cell. Those microtubules that are attached to chromosomes actually shorten and pull those chromosomes in, almost like you're reeling in an anchor rope or something, just pulling it in. So this pulls the chromosomes to either pole, and it elongates the cell. The reason that the cell needs to elongate is that it needs to divide. So once those chromosomes are pulled to either pole, we enter telophase, which is the last part of mitosis. And at the same time, the cell also enters cytokinesis, which is the next phase, or the next stage of mitosis. It's part of the meiotic phase, but it's not part of the nuclear cell division. In telophase, the nucleus begins to reform. Those microtubules are pulled in and broken down, and the chromosomes start to decondense. So your chromosomes unpack. At the same time, if we're talking about an animal cell, we get what's called a cleavage furrow. So basically, another set of microtubules comes in and pinches this cell in half, almost like it's pulling a drawstring. And so it partitions that cytoplasm, it pinches the cell membrane in there, in the center, until the cell membrane is pinched together in the middle and one cell buds off from the other. So it's that pinching and partitioning that is the cytokinesis. And once cytokinesis is finished, you now have two daughter cells. So let's look at cytokinesis a little more closely. So at this point, telophase is occurring or has finished up. Our chromosomes are decondensing because they have to get ready for G1 phase, where they're going to do their job and express their genes. And then the nucleus is reforming. So we have two fully formed nuclei, one in each of these daughter cells. And you can see there that as this takes place, we get this cleavage furrow. So the cell sort of, the microtubules pinch the cell down the center and basically split it into two cells. I like to sort of think about this as like if you watch somebody make a balloon animal and they twist one end of the balloon and it pinches in. Obviously that's not fully cell division, but that's sort of what's happening there. It's pinching that cell or that balloon into two. So those microtubules, these are special ones called microfilaments, pinch in. All of the organelles and the cytoplasm are partitioned. And finally, once telophase and cytokinesis is done, we get our two daughter cells. So the cleavage furrow occurs in animal cells or other cells, something like a protist that doesn't have a cell wall. Some protists do, but if an animal has a cell wall, it presents a problem. You can't pinch in a cell wall. So in plants, cell walls are made up of cellulose, and we said that cellulose was a really strong, almost mat-like fiber, and you can't just pinch that in half. So instead, plants perform cytokinesis by something called a cell plate. So instead of a cell furrow, we have a cell plate. In the production of the cell plate, we get vesicles that have budded off from the Golgi apparatus, and they move to the center or the equator where the chromosomes were lined up. So they line up at the metaphase plate. And this series of vesicles form the beginning of a cell wall. So they actually form this, this almost temporary structure. But those vesicles begin piling up, and eventually you get enough piled up in the center that cell plate meets the cell wall on either end of the cells and has divided that original cell into two cells. So instead of pinching that cell wall together, we get a divider put up. So we essentially build a new wall between those daughter cells. This is a picture of plant cell mitosis. We'll actually look at this in our lab, but if you look closely, you can see some of the different phases. So over here we probably have prophase. Those cells are, or chromosomes are pretty condensed in the cell, in the nucleus. Most of these cells are actually in interphase because the plant spends most of its time in interphase. We have, this is a good picture of anaphase. You can see those chromosomes being pulled apart. This one here is probably late metaphase, early anaphase. They're probably were lined up at the pole, but maybe not anymore. This one might be metaphase, not quite sure about that. Over here we've got, you can actually see the cell plate being formed. So this is telophase. Those chromosomes are forming into a nucleus again, decondensing, and then you have that cell plate forming. Over here we have another one, a telophase with the cell plate forming.